Hey everyone, in this video you'll learn how to count the number of chromosomes, DNA molecules, and the number of copies of each gene that are present during each phase of meiosis. And at the end of the video, remember to fill out the learning guide as well. Now in this video, we'll address one question. And our question is, how does the number of chromosomes, DNA molecules, and gene copies change throughout meiosis? Here we see a figure that shows meiosis 1 and 2. In this illustration, there are 3 chromosome types: a large one, a medium-sized one, and a small one. Don't be confused by the different colors. They just serve to illustrate that the homologous chromosomes could have different alleles. These two are 2 homologous chromosomes. Now, let's see how the number of chromosomes, DNA molecules, and gene copies changes throughout meiosis. Note that this particular diagram doesn't show prometaphase 1 and 2, which is fairly common. Remember that the person that does the work does the learning. So instead of me just telling you the answers, in a second I'll ask you to fill in these numbers yourself. These types of questions are common on exams and quizzes. Keep in mind that I'm asking for the number of each of these things per cell. We'll consider these cells in telophase 1 to be 2 separate cells, as they will be by the end of telophase 1 and cytokinesis. And here, after telophase 2 and cytokinesis, there are 4 separate cells. So at this point, pause the video and fill in the number of chromosomes, DNA molecules, and the number of copies of each gene in each phase of meiosis 1 and meiosis 2. Okay, if you attempted to determine the number of chromosomes, DNA molecules, and gene copies per cell for all of these phases, congratulations, you are a diligent student. And remember, getting them wrong at this point is an excellent way to learn this. You all have heard me say this before, but making mistakes is an important part of learning. And if it seems like I know this stuff pretty well, it's because I've made a lot of them. Okay, so in prophase 1, we can count 1, 2, 3, 4, 5, 6 chromosomes. We see 3 tetrads, each containing 2 chromosomes. So there are 6 chromosomes total in prophase 1. At this point, each chromosome has 2 DNA molecules, or sister chromatids. So there are 12 DNA molecules in this cell. Now for the number of copies of each gene per cell. Let's try to visualize one gene. Let's say the largest chromosome type has the A gene. This chromosome is made of 2 sister chromatids, which of course have the same genes. The other homologous chromosome in the tetrad also has 2 sister chromatids, which will also have the same genes as the first chromosome. So there are 4 copies of this gene, and indeed there are 4 copies of all genes present in this cell. Another way to think about it is each tetrad has 4 chromatids, and these chromatids have the same genes, so there are 4 copies of each gene. Now, during metaphase 1, the tetrads line up near the metaphase plate, but nothing else really happens to the chromosomes, so we still have 6 chromosomes, 12 DNA molecules, and 4 copies of each gene. During anaphase 1, the homologous chromosomes are pulled apart, but the sister chromatids stay together. So we still see 1, 2, 3, 4, 5, 6 chromosomes. Each chromosome still has 2 chromatids, or DNA molecules, so there are still 12 DNA molecules and still 4 copies of each gene. During telophase 1 in cytokinesis, the cell splits, with each cell getting half of the chromosomes the anaphase cell had. So these 2 cells have 3 chromosomes each. Or if we have access to a diagram, we could also just count them. 1, 2, 3. Each chromosome has 2 DNA molecules, so each cell now has 6 DNA molecules total. And since we only have 1 chromosome of each type, and each has 2 chromatids, there are now only 2 copies of each gene in these cells. 2 A genes in this cell, and 2 A genes in this cell. I'm not worrying about what alleles these genes are because I don't need to know that to answer these questions. During telophase 1, the chromosomes are decondensing, but each chromosome still consists of 2 chromatids during telophase and cytokinesis and during interkinesis, which is not shown here. So we still have 3 chromosomes in each of these cells. During prophase 2, the chromosomes condense, but they don't otherwise change, so we still have 3 chromosomes, 6 DNA molecules, and 2 copies of each gene in each cell. In metaphase 2, the chromosomes line up, but don't otherwise change, so we still have 3 chromosomes, 6 DNA molecules, and 2 copies of each gene in each cell. During anaphase 2, the sister chromatids of each chromosome are now pulled apart. So the number of chromosomes in each cell doubles, because each chromatid is now considered its own chromosome. So we now have 6 chromosomes in each cell. The number of DNA molecules is also 6, because now each chromosome is made up of only 1 DNA molecule. Each cell still has 2 copies of each gene. Finally, during telophase 2 in cytokinesis, each cell splits into 2 cells, giving half of its chromosomes to each daughter cell. So each of these cells has 3 chromosomes, 1 of each type, and since they only have 1 of each chromosome type, and each is only 1 DNA molecule, they only have 1 copy of each gene. For the same reason, each cell has only 3 DNA molecules, 1 of each type, so these cells are in fact haploid. Sometimes the amount of DNA in a cell is not measured by the number of DNA molecules, but instead by the mass of DNA present in the cell. DNA in eukaryotic cells is often on the order of a few picograms. A picogram is 10 to the minus 12 grams. The mass of DNA in a cell is proportional to the number of DNA molecules in that cell. So if you know the mass of DNA in one phase, like G1, you can determine the mass of DNA in the other phases by knowing how the number of DNA molecules changes throughout the cell cycle and through the phases of either mitosis or meiosis. For example, if we know that there is 1 picogram of DNA in a prophase 1 cell, then there will also be 1 picogram of DNA in metaphase 1 and anaphase 1 cells, and 0.5 picograms in each cell after telophase and cytokinesis. Same for prophase 2, metaphase 2, and anaphase 2, with each cell after telophase 2 and cytokinesis having now 0.25 picograms per cell. The amount of DNA per cell tracks with the number of DNA molecules. In this example, we were looking at a 2n equals 6 species, but keep in mind that humans have 46 chromosomes in G1, so the chromosome number would change accordingly throughout meiosis. Okay, so at this point you should be able to determine the number of chromosomes, DNA molecules, and gene copies that are present at each phase of meiosis. And if you don't feel like you can, as always, feel free to watch the video again. Now, in the next few videos, we'll take a look at how meiosis and fertilization are a large part of why all of us are different from each other, at least genetically speaking. See you all then.